megaspore. This megaspore is formed when one of the nucellus cells, towards its micropylar end, gets differentiated into a megaspore mother cell, which can be easily distinguished from other cells due to its large size, dense cytoplasm, and prominent nucleus. The megaspore mother cell undergoes meiosis to form 4 megaspores which arrange themselves in a linear tetrad. This process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Of the 4 megaspores, usually the one near the chalazial end becomes functional, whereas the other 3 near the micropylar end degenerate. The functional megaspore enlarges and simultaneously undergoes mitotic division to form an embryo sac. This type of formation of the embryo sac from a single megaspore is known as monosporic development. While undergoing these mitotic divisions, the nucleus of the functional megaspore first divides to form 2 nuclei, which move to the 2 opposite ends of the embryo sac. This is the 2-nucleate stage of the embryo sac. The mitotic divisions continue and result in the formation of the 4-nucleate and later the 8-nucleate stages of the embryo sac. A mature embryo sac thus has 8 nuclei after 3 mitotic divisions, which are arranged in a group of 4 at each end of the embryo sac. Interestingly, the mitotic divisions that occur in the megaspore's nucleus are free nuclear, which means the division of the nuclei doesn't immediately trigger cytoplasmic division and cell wall formation. Instead, we see the formation of cell walls only after the 8-nucleate stage, due to which 6 of the 8 nuclei organize into cells. The 2 remaining nuclei, called polar nuclei, migrate to the center of the embryo sac. Thus, we find the embryo sac to be in 7-celled and 8-nucleate stage, with 6 cells at the poles and a large central cell with 2 nuclei in the center. Meanwhile, the 6 cells organize to form the typical structure of the embryo sac, which is now ready for fertilization. The 3 cells present at the chalazial end, for instance, group together to form the antipodal cells, which do not have any specific function. Whereas the 3 cells at the micropylar end group together to form the egg apparatus. While one of the cells functions as an egg or the female gamete, the other 2 cells are called synergids. If you observe the micropylar tip of the synergids, you will notice special cellular thickenings called a filiform apparatus that guides the pollen grains into the synergid. The synergids, as well as other cells, form the embryo sac, the female gametophyte born inside the pistil.